Now there's this algorithm called MCL, the Markov Clustering Algorithm. You'll see it here. Really all the thing that you really need to worry about are two things. First of all is the inflation value, and we set it at this value of 2.2, and for most purposes I would stick at that. Okay, so the inflation value, well, let me just run it first of all, and then you'll understand what the clustering algorithm does and how we can use it. Okay. So you see now we have color. We have colored our graph, and we have colored our graph with put things into groups, and you can see that the color really kind of represents where they are within the graph. So all of these guys here are kind of purple, yeah, and we've got kind of green things there, brown, purple, that there is a relationship there. So what this algorithm does is it looks for the structure and then says anything which is highly connected we're going to actually call as a cluster. Okay. Now there's the first thing that I should say, not the layout, I want the MCL. This value here, smallest cluster allowed. Now in these type of graphs we should probably make that 1. So although it's counterintuitive, you can have a cluster of 1. Okay, something can be clustered on its own. So if I change that now, and you'll notice that all of these guys here are blue, and if I just select all the other blue ones, and the reason is that I had, when I clustered it last time, the smallest cluster allowed as 3, therefore anything that was less than 3 was given what is a class called no class. Okay, it wasn't part of a cluster. So now that I've changed that to 1, when I cluster it again, we'll see that everything has a color. Okay, so now I've allowed these clusters here based on only just 2 nodes. Does that make sense? All right. Now, clustering is really important in this world because what it allows us to do is to take very large graphs and to break it into manageable chunks. And manageable chunks of things what are related. Okay. Now, the other thing I'll mention here is, in terms of the MCL, is that if we increase this value, let's go up to a bit 10. Okay, doesn't really matter. And I run it again, you'll notice that that group of things which were all green and called the same cluster before have now been shattered into smaller clusters. Okay, so what was considered as a threshold of 2.2 to be one cluster is now considered to be lots of little clusters, and so I've fragmented it up into smaller chunks. And likewise, if I go below that value and I'll go down to 1.2 or 1.4 and run it, okay, it's now done a less stringent clustering. So clustering, there's no such thing as a right and wrong clustering. There is no setting. The way I think about graphs is that I want to capture in the clustering something about the relationships that I can see based on just visualizing the structure. Okay, so let's go back to, so having now clustered a graph, your nodes are in a, it's a bloody screen. I think at the end here. So when you cluster a graph, you put nodes into a class, and that class is their cluster, and their cluster is defined based on what the MCL setting. So you can see here that through going through this graph, I now have grouped all of my nodes in my graph into 3 separate classes: MCL 2.2, MCL 10.4, and MCL 1.4. And I can go back to those at any one time because these are their groupings now. And I put this here and apply, and I've got back to my 2.2 clustering. Okay? So why is this clustering important? Because it allows us to inspect what these clusters are. So either by pressing this little cog key, so this cog key here, the class viewer, is one of the most important tools within BioLayout. We are going to just go on here, and we can say find by class. Okay, so our class is currently set at clustering at 2.2, and we want to look at cluster 1. So when you select a cluster, you essentially see that it now has this wire framework around it, and that wire framework says that they're selected. That's what you're viewing at within the class viewer. And here you can see all of the things which are what are in cluster 1. Okay, so cluster 1 contains all the adrenal tissue, the B cells, the bladder, CD4, CD8s, embryonic tissue. And actually one of the reasons that, well, we'll come back to this in a second, but if we go to the next, so let's just look at this, let's go to the next class. And now what we see is that it's grouped all the brain samples together. So in this set of samples, I think there's something like 16 different brain areas of the mouse, and it has grouped them all together as a cluster. Then digits, epidermis, tongue, everything with a dermal layer, basically. And as we walk through there, there's the bone and bone marrow, small and large intestines, fertilized egg and oocyte, brown fat, heart, lung, okay, stomach, etc., etc. So we've now been able to take this graph and break it into chunks, and that what we've been able to see is explore this graph for what those in those chunks and see that all is good, that samples are clustered together in a way. Now, if you want to sort of potentially, now this is the kind of graph that I would often go in 2D for and look in 2D, because obviously in 3D, if you want to make a presentation of this, then, you know, the great thing about 3D is that when you're looking at it, live, you can do all of that, yeah. But when you want to present it as a piece of paper, then actually it's sometimes better to go into 2D. And so if we go back here and press recluster it, now I can take this graph and we can put all things together here, and I can make that up as a nice labeled diagram, put it into a paper, put it back to 3D, and yep, I can now annotate that and show to people how my data clustered together, and isn't it nice that everything clusters around. So just one other thing before we go for coffee. When I laid this graph out, I used quite a low correlation value. So if you press this button here, open the last graph, what it'll do is load the last graph that you had, which is this one, of course. I'm going to go back and transpose it again, say OK. Come back to this graph. So we looked here at 0.7. I'm going to take this up, say, midway to 0.85. And what you'll see, I've now gone from 0.7, where I had 993 edges. And it has to say, if I'd taken this at 0.5, what would eventually happen is you'd end up with something that looks like a blastocyst. All the samples would be grouped together and formed into one mass, because your threshold that you'd use to separate them is basically one that means that there is no separation and everything is conjoined with everything else. So if you lower this down further, you'll basically just get a ball of everything. So what I'm going to do now is actually take it up higher and carve off a whole load of edges and show more of the structure. So quite often what we're looking for, not so much in these sample graphs, but in the bigger graphs, is actually a point where we have all the nodes, but we've lost a lot of edges. We're trying to trim out edges. So let's take it at 0.85. And you can see now that that graph is fragmented, okay? Because I've cut away all the edges which were loose connections. So tissues which were kind of related but not that related have now been chopped away, and that have now, well, let me just, you'll probably see things like this, I'm guessing is the embryonic tissue. Oh, that's the C. So this group of cells before was in cluster 1. But now that I've increased the threshold, what it's actually done is nicely separate out all the lymphoid-based tissues and cell types and call them a separate group, which we can see as a separate group, which before, well, we could see it in the local structure, but it wasn't as obvious as that now. Okay? Does that all make sense? I'm assuming these are the embryonic samples then. Oh yeah, there we are. So all the embryos again separated away from there. Right, so I think lesson 1 is potentially complete, and I hope we're started now. But the point about this approach for looking at data is that your first thing you want to do is a sanity check. Is all things related? Is there any gross differences between samples that mean that your samples are separating out in a way that suggests there's a big difference between them? Now, obviously, this is an unusual experiment where you're actually taking a whole atlas. Generally, our biological experiments are a lot more focused than this, but the question is still obviously the same. Is there any overall grouping within the data that separates out samples? It's your first indication. So whatever you see at this level will be reflected at the gene level. Okay? If there is, you will find there's a bunch of genes that are only expressed in the embryo. You'll find there's a bunch of genes that are only expressed in the B cells, T cells, lymphoid. It's the structure that this is a gross overview of the underlying structure that you'll find when you start looking at the genome. So I think this is a good place to stop. Grab a quick coffee